You know, the rules of inheritance were first worked out by Gregor Mendel. He was an Augustinian monk.
This is Dr. George W. Beadle of the California Institute of Technology. Born and raised on a Nebraska farm, he first studied the then new sciences of heredity and evolution at the University of Nebraska's College of Agriculture in 1924. The work Dr. Beadle has done since then has greatly added to man's knowledge of the process by which tiny particles in living cells control the forms that life will take. The tiny particles are called genes. The science of their study is called genetics. Dr. Beadle's work in this science won him a share in a Nobel Prize in 1958.
You already know that each of us starts development as a tiny, almost microscopic single cell, the fertilized egg. Here it is, seen through a microscope. It's about the size of the point of a dull pin. Here's another egg, about the same size as the other. But this isn't a human egg. It's the egg from which a sea urchin develops. How does each of these egg cells know what it's supposed to develop into? For instance, how does the human egg cell know that it's supposed to develop into a human being? It knows because the cell contains an elaborate set of directions, or specifications, for making people, not sea urchins. These specifications determine what color our hair will be, whether we will be male or female. To a large extent, they determine what we will look like, and they determine many thousands of important things about the way we will grow and develop. Most of these specifications are contained in the nucleus, the microscopic central sphere of the egg cell, located in tiny bodies called chromosomes. The specifications we call genes. I'm now going to ask 5 questions about these specifications and then tell you what we think the answers might be. First, how are the specifications transmitted from one generation to another? I'm going to illustrate with some mice. This mouse has genes in each of his cells that contain specifications that say he will produce brown pigment. This one has specifications that say she will not produce pigment, and the lack of pigment leaves her with white fur. If these 2 mice mate together and produce offspring, each will contribute specifications through egg and sperm cells to the offspring, the mother for no pigment, the father for brown pigment. The sons and daughters will therefore have 2 sets of specifications concerning the color of their fur. Now, how are the specifications written? This mouse has specifications that say, in effect, I cannot produce pigment. This woman is an albino. She also has genes carrying specifications that say, I cannot produce pigment. Now we know we don't have words and sentences in our cells written in the 26 letters of our alphabet, but nevertheless the information is there. We can write, I cannot produce pigment, in a simple code using only 2 symbols, dots and dashes. To someone who knows the Morse code, this says, I cannot produce pigment. The specifications for the development of the fertilized egg cell are written in a code that uses 4 symbols. The code is written in long spiral staircase-like molecules of a substance called deoxyribonucleic acid. It's called DNA for short. Now this is a model of a molecule of DNA. DNA molecules are made of a large number of atoms and are quite complicated. But they are made of smaller, less complex units, and it's the way these smaller units are arranged that determines what the code is saying in each particular piece of DNA. Let's look at a simplified diagram of a DNA molecule. If we were to untwist the spiral, the basic shape of a DNA molecule would be somewhat like a ladder. The long supports are made up of connected subunits of sugar and phosphates. We'll indicate these with the letters S and P. The rungs, or connecting pieces, are 2 units each and are of 4 different kinds. We can simply label them A, T, C, and G. Each connecting rung of the DNA molecule is made up of 2 of them. An A is always combined in a rung with a T, and a C is always combined with a G. However, they can be combined in either direction, and the rungs can be arranged in any order. Now, the Morse code has 2 symbols, dots and dashes, and the order in which they appear indicates what letter they stand for. The DNA code has 4 symbols, and the order in which they are strung together is the code that spells out the specifications that tell the egg cell how to develop. Now, of course, this code doesn't form letters and words, but it does record information that controls and directs the development of an egg into a fully grown organism. Located on one of the chromosomes of this albino woman is a piece of DNA that says in some way in its 4-symbol code, "I cannot produce pigment." It's been estimated that it takes from 1,000 to 10,000 of these code units arranged in a particular order to encode such a specification. Just imagine how many different specifications it takes to determine how you will develop. There are some for the color of your hair, others for the color of your eyes, others for your blood type. Each of us has perhaps 100,000 separate specifications contained in the fertilized egg cell from which we develop. Collectively, these 100,000 specifications add up to a kind of a recipe for us. They say how to make us out of the egg cell, given time, food, proper temperature, and so forth. If we expressed all this information in English, it would fill about 1,000 volumes. Yet all this information is contained in that tiny microscopic sphere that is the nucleus of the fertilized egg cell. This fertilized egg cell is the beginning of a human life. Now let's examine, how does it develop into a human being? Do you know what mitosis is?
Mitosis is the way a cell is divided into 2 cells. It's the way new cells are made.
That's right. It's through the process of mitosis that we develop from one fertilized egg cell to an organism with millions of cells. Here we see a cell divided by the process of mitosis. The time is sped up by the camera. Actually, it takes much longer. You'll study mitosis in some detail later. But what I want you to consider now is what happens to the specifications in the nucleus of the cell when the cell divides. What do you think happens?
Do they divide some way too?
That's absolutely right, they do. When a cell divides, the 2 new cells contain the same specifications the parent cell contained. That means that the specifications, that 1,000 volumes worth of information you remember, have been reprinted into every nucleus of every single cell in your body. And remember, you have millions of them. The reprinting of the specifications is directly tied in with the structure of the DNA molecules on which the specifications are coded. Several kinds of experiments indicate that the reprinting occurs by separation of the molecule into 2 halves, followed by the formation of new partners by each half. Now there is a complete DNA molecule for the nuclei of each of the 2 new cells. We'll look at that more closely and see exactly what happens. When the halves of the DNA molecule separate, they separate between the 2 units of the connecting bonds along the molecule. In the cells in which this is happening, and it's happening in your cells right now, there are many single subunits made from the food we eat. When these come into contact with their proper partners, they are held in place. And A only joins with a T. a C with a G. Now a complete new molecule is formed, identical to the parent molecule. The coded specification it carries has been reprinted just as it was, because the sequence of the code symbols in the half that came from the old molecule controls exactly the sequence in the new half that is formed. This is a molecular model of the G subunit of the DNA molecule. It's magnified 100 million times. Imagine that it's part of one of the halves left after the DNA molecule has divided. This is a model of a T subunit. Imagine that it's free in the cell. If it tries to join with a G subunit, it won't hold, doesn't fit. But when a C subunit comes along, it fits perfectly. From one DNA molecule carrying its coded specification, two exactly like the original are formed. These go into the two daughter cells that result from the division of the parent cell. In terms of molecules, this is the basis of all biological reproduction. This reprinting of the specifications from cell to cell is done with high precision. There's perhaps only one typographical error, that is only one misplaced symbol, for each reprinting of the thousands of DNA molecules in a single cell. Remember, the information encoded in these molecules is equivalent to 1,000 printed volumes. Reprinting that information amounts to a typist copying 1,000 volumes and making only one significant typographical error. What about the occasional mistakes in DNA specifications? Mistakes in reprinting, like typographical errors, or mistakes that occur in other ways. We call these mistakes mutations. Usually, mutations give rise to wrong recipes and are harmful. For example, if there's a mistake in the DNA specifications for a hemoglobin protein, the hemoglobin made according to this specification will be defective. This is a cell with normal hemoglobin, the red pigment molecules of your blood that are necessary for carrying oxygen through the circulatory system. This is a cell with defective hemoglobin. It's called sickle cell hemoglobin because the red cells containing it are distorted, often in the shape of a sickle. This defective hemoglobin occurs in man when there is a mistake in the DNA instructions for producing hemoglobin. Such cells come to an untimely end, and the bearer of them suffers from sickle cell anemia and often does not live to maturity. Now, I don't want you to think that all mutations are unfavorable. As a matter of fact, most of them are. But you'll learn that the entire process of evolution has depended upon favorable mutations. We have evolved from one-celled creatures of billions of years ago through errors in DNA specifications that have improved the recipe for the organism in which they occurred. This is the principle behind the development of hybrid seed and livestock. And it's the principle nature has used in the development of such complex creatures as ourselves, able to cope with our environments in a remarkably satisfactory way. I hope I've aroused your curiosity about these amazing DNA molecules that carry your biological specifications. If you want to learn more about them, and there is much more no one yet knows, I strongly urge to learn as much as possible about the chemistry of simpler molecules. Such study will be well worthwhile, because in a real sense, DNA holds the secret of life itself.